Pulmonary function tests. Chapter 121. Introduction. Pulmonary function tests or lung function tests are useful in assessing the functional status of the respiratory system both in physiological and pathological conditions. Lung function tests are based on the measurement of volume of air breathed in and out in quiet breathing and forced breathing. These tests are carried out mostly by using spirometer. Types of lung function tests. Lung function tests are of two types: static lung function tests, dynamic lung function tests. Static lung function tests. Static lung function tests are based on volume of air that flows into or out of lungs. These tests do not depend upon the rate at which air flows. Static lung function tests include static lung volumes and static lung capacities. Dynamic lung function tests. Dynamic lung function tests are based on time, i.e. the rate at which air flows into or out of lungs. These tests include forced vital capacity, forced expiratory volume, maximum ventilation volume and peak expiratory flow. Dynamic lung function tests are useful in determining the severity of obstructive and restrictive lung diseases. Lung volumes. Static lung volumes are the volumes of air breathed by an individual. Each of these volumes represents the volume of air present in the lung under a specified static condition, specific position of thorax. Static lung volumes are of four types: tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume. Tidal volume. Tidal volume, TV, is the volume of air breathed in and out of lungs in a single normal quiet respiration. Tidal volume signifies the normal depth of breathing. Normal value: 500 ml, 0.5 L. Inspiratory reserve volume. Inspiratory reserve volume, IRV, is an additional volume of air that can be inspired forcefully after the end of normal inspiration. Normal value: 3,300 ml, 3.3 L. Expiratory reserve volume. Expiratory reserve volume, EVR, is the additional volume of air that can be expired out forcefully, after normal expiration. Normal value: 1000 ml, 1 L. Residual volume. Residual volume, RV, is the volume of air remaining in lungs even after forced expiration. Normally, lungs cannot be emptied completely even by forceful expiration. Some quantity of air always remains in the lungs even after the forced expiration. Residual volume is significant because of two reasons: it helps to aerate the blood in between breathing and during expiration. It maintains the contour of the lungs. Normal value: 1200 ml, 1.2 L. Lung capacities. Static lung capacities are the combination of two or more lung volumes. Static lung capacities are of four types: inspiratory capacity, vital capacity, functional residual capacity, total lung capacity. Inspiratory capacity. Inspiratory capacity, IC, is the maximum volume of air that is inspired after normal expiration, and expiratory position. It includes tidal volume and inspiratory reserve volume, Fig 121.1. IC equals TV plus IRV. Equals 500 plus 3300 equals 3800 ml. Vital capacity, VC. Vital capacity, VC, is the maximum volume of air that can be expelled out forcefully after a deep, maximal, inspiration. VC includes inspiratory reserve volume, tidal volume, and expiratory reserve volume. VC equals IRV plus TV plus of. Equals 3300 plus 500 plus 1000 equals 4800 ml. Vital capacity is significant physiologically and its determination is useful in clinical diagnosis as explained later in this chapter. Functional residual capacity. Functional residual capacity, FRC, is the volume of air remaining in lungs after normal expiration, after normal tidal expiration. Functional residual capacity includes expiratory reserve volume and residual volume. FRC equals of plus RV equals 1000 plus 1200 equals 2200 ml. Total lung capacity. Total lung capacity, TLC, is the volume of air present in lungs after a deep, maximal, inspiration. It includes all the volumes. TLC equals IRV plus TV plus of plus RV. Equals 3300 plus 500 plus 1000 plus 1200 equals 6000 ml. Measurement of lung volumes and capacities. Spirometry is the method to measure lung volumes and capacities. Simple instrument used for this purpose is called spirometer. Modified spirometer is known as respirometer. Nowadays plethysmograph is also used to measure lung volumes and capacities. Spirometer. Spirometer is made up of metal and it contains two chambers, namely outer chamber and inner chamber, Fig 121.2. Outer chamber is called the water chamber because it is filled with water. A floating drum is immersed in the water in an inverted position. Drum is counterbalanced by a weight. Weight is attached to the top of the inverted drum by means of string or chain. A pen with ink is attached to the counterweight. Pen is made to write on a calibrated paper, which is fixed to a recording device. Inner chamber is inverted and has a small hole at the top. A long metal tube passes through the inner chamber from the bottom towards the top. Upper end of this tube reaches the top portion of the inner chamber. Then the tube passes through a hole at the top of inner chamber and penetrates into outer water chamber above the level of water. A rubber tube is connected to the outer end of the metal tube. At the other end of this rubber tube, a mouthpiece is attached. Subject respires through this mouthpiece by closing the nose with a nose clip. When the subject breathes with spirometer, during expiration, drum moves up and the counterweight comes down. Reverse of this occurs when the subject breathes the air from the spirometer, i.e. during inspiration. Upward and downward movements of the counterweight are recorded in the form of a graph. Upward deflection of the curve in the graph shows inspiration and the downward deflection denotes expiration. Spirometer is used only for a single breath. Repeated cycles of respiration cannot be recorded by using this instrument because carbon dioxide accumulates in the spirometer and oxygen or fresh air cannot be provided to the subject. Respirometer. Respirometer is the modified spirometer. It has provision for removal of carbon dioxide and supply of oxygen. Carbon dioxide is removed by placing soda lime inside the instrument. Oxygen is supplied to the instrument from the oxygen cylinder, by a suitable valve system. Oxygen is filled in the inverted drum above water level and the subject can breathe in and out with instrument for about 6 minutes and recording can be done continuously. Spearogram. Spirogram is the graphical record of lung volumes and capacities using spirometer. Upward deflection of the spirogram denotes inspiration and the downward curve indicates expiration, Fig 121.3. In order to determine the lung volumes and capacities, following 4 levels are to be noted in spirogram: normal and expiratory level, normal and inspiratory level, maximum expiratory level, maximum inspiratory level. Computerized spirometer. Computerized spirometer is the solid-state electronic equipment. It does not contain a drum or water chamber. Subject has to respire into a sophisticated transducer, which is connected to the instrument by means of a cable. Disadvantages of spirometry. By using simple spirometer, respirometer or computerized spirometer, not all the lung volumes and lung capacities can be measured. Volume, which cannot be measured by spirometry, is the residual volume. Capacities, which include residual volume, also cannot be measured. Capacities that include residual volume are functional residual capacity and total lung capacity. Volume and capacities, which cannot be measured by spirometry, are measured by nitrogen washout technique or helium dilution technique or by body plethysmograph. Plethysmography.it is a technique used to measure all the lung volumes and capacities. It is explained later. Measurement of functional residual capacity and residual volume. Functional residual capacity and residual volume cannot be measured by spirometer and can be determined by three methods: helium dilution technique, nitrogen washout method, plethysmography. 1. Helium dilution technique. Procedure to measure functional residual capacity. Respirometer is filled with air containing a known quantity of helium. Initially, the subject breathes normally. Then, after the end of expiration, subject breathes from respirometer. Helium from respirometer enters the lungs and starts mixing with air in lungs. After few minutes of breathing, concentration of helium in the respirometer becomes equal to concentration of helium in the lungs of subject. It is called the equilibration of helium. After equilibration of helium between respirometer and lungs, concentration of helium in respirometer is determined, Fig 121.4. Functional residual capacity is calculated by the formula, FRC equals V, C1, C2, C2. Where, C1 equals initial concentration of helium in the respirometer, C2 equals final concentration of helium in the respirometer, V equals initial volume of air in the respirometer. Measured values. For example, the following data of a subject are obtained from the experiment: initial volume of air in respirometer equals 5 L, 5000 ml. Initial concentration of helium in respirometer equals 15%. Final concentration of helium in respirometer equals 10%. Calculation. From the above data, the functional residual capacity of the subject is calculated in the following way: FRC equals V, C1, C2, C2. FRC equals 5,000, 15/100, 10/100. Thus, the functional residual capacity in this subject is 2500 ml. Procedure to measure residual volume. To determine functional residual capacity, the subject starts breathing with respirometer after the end of normal expiration. To measure residual volume, the subject should start breathing from the respirometer after forced expiration. 2. Nitrogen washout method. Normally, concentration of nitrogen in air is 80%. So, if total quantity of nitrogen in the lungs is measured, the volume of air present in lungs can be calculated. Procedure to measure functional residual capacity. Subject is asked to breathe normally. At the end of normal expiration, the subject inspires pure oxygen through a valve and expires into a Douglas bag. This procedure is repeated for 6 to 7 minutes, until the nitrogen in lungs is displaced by oxygen. Nitrogen comes to the Douglas bag. Afterwards, following factors are measured to calculate functional residual capacity. Calculation. Volume of air collected in Douglas bag. Concentration of nitrogen in Douglas bag. By using the data, the functional residual capacity is calculated by using the formula, procedure to measure residual volume. FRC equals C1 times VC2, where V equals volume of air collected. C1 equals concentration of nitrogen in the collected air. C2 equals normal concentration of nitrogen in the air. Measured values. For example, the following data are obtained from the experiment with a subject: volume of air collected equals 40 L, 40,000 ml, concentration of nitrogen equals 5% in the collected air, normal concentration of equals 80% nitrogen in the air. Calculation. From the above data, the functional residual capacity of the subject is calculated in the following way: procedure to measure residual volume. To measure the functional residual capacity, the subject starts inhaling pure oxygen after the end of normal expiration, and to determine the residual volume, the subject starts breathing pure oxygen after forceful expiration. 3. Plethysmography. Plethysmography is a technique to study the variations in the size or volume of a part of the body, such as limb. Plethysmograph is the instrument used for this purpose. Whole body plethysmograph is the instrument used to measure the lung volumes, including residual volume. Plethysmography is based on Boyle's law of gas, which states that the volume of a sample of gas is inversely proportional to the pressure of that gas at constant temperature. Subject sits in an airtight chamber of the whole body plethysmograph and breathes normally through a mouthpiece connected to a flow transducer called pneumotachograph. It detects the volume changes during different phases of respiration. After normal breathing for few minutes, the subject breathes rapidly with maximum force. During maximum expiration, the lung volume decreases very much. But volume of gas in the chamber increases with decrease in pressure. By measuring the volume and pressure changes inside the chamber, volume of lungs is calculated by using the formula, P1 times V equals P2, V, V, where P1 and P2 equals pressure changes. V equals functional residual capacity. Vital capacity. Definition. Vital capacity is the maximum volume of air that can be expelled out of lungs forcefully after a maximal or deep inspiration. Lung volumes included in vital capacity. Vital capacity includes inspiratory reserve volume, tidal volume, and expiratory reserve volume. Normal value. Vc equals IRV plus TV plus of equals 3300 plus 500 plus 1000 equals 4800 ml. Variations of vital capacity. Physiological variations. Sex. In females, vital capacity is less than in males. Body build, vital capacity is slightly more in heavily built persons. Posture, vital capacity is more in standing position and less in lying position. Athletes, vital capacity is more in athletes. Occupation, vital capacity is decreased in people with sedentary jobs. It is increased in persons who play musical wind instruments such as bugle and flute. Pathological variations. Vital capacity is decreased in the following respiratory diseases: asthma, emphysema, weakness or paralysis of respiratory muscle, pulmonary congestion, pneumonia, pneumothorax, hemothorax, pyothorax, hydrothorax, pulmonary edema, pulmonary tuberculosis. Measurement. Vital capacity is measured by spirometry. The subject is asked to take a deep inspiration and expire forcefully. Forced vital capacity. Forced vital capacity, FVC, is the volume of air that can be exhaled forcefully and rapidly after a maximal or deep inspiration. It is a dynamic lung capacity. Normally FVC is equal to VC. However, in some pulmonary diseases, FVC is decreased. Forced expiratory volume or timed vital capacity. Definition. Forced expiratory volume, FEV, is the volume of air, which can be expired forcefully in a given unit of time, after a deep inspiration. It is also called timed vital capacity or forced expiratory vital capacity, FEVC. It is a dynamic lung volume. FEV1 equals volume of air expired forcefully in 1 second. FEV2 equals volume of air expired forcefully in 2 seconds. FEV3 equals volume of air expired forcefully in 3 seconds. Normal values. Forced expiratory volume in persons with normal respiratory functions is as follows: FEV1 equals 83% of total vital capacity, FEV2 equals 94% of total vital capacity. FEV3 equals 97% of total vital capacity. After third second equals 100% of total vital capacity. Some restrictive respiratory diseases like fibrosis of lungs, Fig 121.5. Respiratory minute volume. Definition. Respiratory minute volume, RMV, is the volume of air breathed in and out of lungs every minute. It is the product of tidal volume, TV, and respiratory rate, RR. RMV equals TV times RR. Equals 500 times 12 equals 6000 ml. Normal value. Normal respiratory minute volume is 6 L. Variations. Respiratory minute volume increases in physiological conditions such as voluntary hyperventilation, exercise, and emotional conditions. It is reduced in respiratory diseases. Maximum breathing capacity or maximum ventilation volume. Definition: maximum breathing capacity, MBC, is the maximum volume of air, which can be breathed in and out of lungs by forceful respiration, hyperventilation, increase in rate and force of respiration, per minute. It is also called maximum ventilation volume, MVV. MBC is a dynamic lung capacity and it is reduced in respiratory diseases. Normal value. In healthy adult male, it is 150 to 170 L/minute and in females, it is 80 to 100 L/minute. Equals 160 L. Peak expiratory flow rate. Definition: peak expiratory flow rate, PEFR, is the maximum rate at which the air can be expired after a deep inspiration. Normal value. In normal persons, it is 400 L/minute. Measurement. Peak expiratory flow rate is measured by using right peak flow meter or a mini peak flow meter. Significance of determining PEFR. Determination of PEFR rate is useful for assessing the respiratory diseases, especially to differentiate the obstructive and restrictive diseases. Generally, PEFR is reduced in all type of respiratory disease. However, reduction is more significant in the obstructive diseases than in the restrictive diseases. Thus, in restrictive diseases, the PEFR is 200 L/minute and in obstructive diseases, it is only 100 L/minute. Restrictive and obstructive respiratory diseases. Diseases of respiratory tract are classified into two types: restrictive respiratory disease, obstructive respiratory disease. These two types of respiratory diseases are determined by lung functions tests, particularly FEV. Restrictive respiratory disease. Restrictive respiratory disease is the abnormal respiratory condition characterized by difficulty in inspiration. Expiration is not affected. Restrictive respiratory disease may be because of abnormality of lungs, thoracic cavity, and nervous system. Obstructive respiratory disease. Obstructive respiratory disease is the abnormal respiratory condition characterized by difficulty in expiration. Obstructive and respiratory diseases are listed in Table 121.1.